So now that we know that replication occurs via a semi-conservative process, let's start talking about what actually occurs at the molecular level. So I'm going to do this in two parts. I'm going to do a general overview, talk about some of the components, and then we will move into the actual mechanism and some of the sort of problems that arise when you attempt to replicate a DNA molecule and how cells overcome those. So I'm not sure yet if this is going to be one or two videos. I might make it two just so each one's a little bit shorter. We'll see how it goes. All right, so to start DNA replication, DNA strands must dissociate. So remember that dissociate means to pull apart. And then after they pull apart, they can actually re-anneal or come back together. So one of the things that we're going to see is an enzyme that actually holds them apart. Now, this dissociation and annealing is really important. We'll sort of see this many times as we're talking about replication and transcription and some different laboratory techniques. But in order to dissociate a molecule, you can either heat it if you're in a laboratory in order to cause it to pull apart. That heat will interrupt the hydrogen bonds in between. And you might do this for PCR sequencing, those types of things. If you are in a cell actually performing DNA replication, you are going to use proteins. So there's a couple of new terms here. In vitro means in a test tube is what it really means. It means experimentally. And in situ means in real life, like what actually happens in the real process. So in a lab, we're going to use heat. And in a cell, we're going to use enzymes or protein molecules. So the first thing we have to do is pull them apart. And in the process of transcription, after we're done taking out our little bit of DNA information, we're actually going to re-anneal our strands. So that's pretty cool. The hydrogen bonds can actually come back together. So this process of DNA replication or transcription, neither one is sort of a permanent block. We can actually put things back together if we need to. So overall view of replication. There are sort of two major steps to this. To start off replication, two strands of DNA are going to be pulled apart. Now this is going to happen using an enzyme called helicase. Now we call it helicase because it separates our strands, essentially sort of unraveling or unwinding the double helix. Now we have another molecule that is also involved with this double helix. In a few minutes, we'll talk about that. But first we need to pull apart our strands using helicase. And then each strand is now open to act as a template for our new strand of DNA. And this process of making the new strand of DNA is carried out by an enzyme called DNA polymerase. So let's look at this word. Ase generally means enzyme or a process. Polymer is a molecule made up of subunits. So we are going to be making DNA out of individual subunits via an enzyme that's going to make this happen. So there's our DNA polymerase. This is the molecule that we're going to use to build our DNA. So up here we have our parental strands. Our helicase molecule is going to pull it apart, essentially interrupting those hydrogen bonds. And then two proteins, these are large proteins called DNA polymerase, are going to come in and bind to both of our new strands. And they're going to facilitate the addition of new nucleotides. So those DNA polymerase molecules, I keep saying molecules, those DNA polymerase complexes are going to move along our strand, replicating on both sides at the same time. Now remember that in order to add a new nucleotide to our DNA strand, we have to have a free 3' end. Now, DNA polymerase works by adding the next complementary base to that 3' hydroxyl group. Now, this is important because we can only add to the free 3' end. So we have a free 3' end here, and we have another one here. We just don't have any unpaired nucleotides drawn there. But we should have a free 3' end on either side of our strand, really. The other thing that's important is that DNA polymerase must have a free 3' end. So if we have a molecule like this, that doesn't have a complementary strand attached or doesn't have an end of that complementary strand available, DNA polymerase, which was that big green blob on the previous page, DNA polymerase cannot, I should have made that red, cannot bind if it does not have a free 3' end. But if we have another strand, if we have the other end of our strand, draw it over here, and we have our free 3' end, then DNA polymerase can absolutely add a new base pair there. So we have to have a 3' end sort of hanging off a complementary strand that's already present, and that's going to be important in just a minute. Keep that in mind. So there are a bunch of different DNA polymerases that are involved in this replication process. And the polymerases in bacteria are a little bit different than the polymerases in eukaryotes, although they all perform sort of the same jobs. Remember I said in one of the lectures, I don't remember which one, that these processes of replication, transcription, translation are very, very similar in bacteria and eukaryotes. And so we often study bacteria as an easier mechanism to learn from. Now in eukaryotes, they just tend to be more complicated. There tend to be more moving parts. And so if we can look at a bacterial model, it's much, much easier. I have this big list of polymerases here. I want you to know the bacterial polymerases. You don't need to memorize the eukaryotic polymerases. I want you to sort of have a general idea of the jobs that polymerase does in eukaryotes, but you don't need to know all of these specific ones. I do want you to know the bacterial ones though, because there's only a couple and they all play different, very important jobs. So DNA polymerase 3, that's this Pol 3 here, does most of the replication. So it's the one that we're going to sort of be talking about most of the time, indicated by that big green blob that's going to move along our strands and add new nucleotides as we go. Now, DNA polymerase 1 does something called processing Okazaki fragments. So we'll see in a minute that one of our strands is actually going to be made in little pieces, and those fragments have to be linked together. So this guy, polymerase 1, is going to link our fragments, or basically fill in little gaps between our fragments. And then DNA polymerase 2, 4, and 5 are going to be working on repair. So they're basically going to be proofreading and correcting. And then we're actually not going to worry about mitochondrial DNA synthesis. I want to note one specific important polymerase. This is the Taq polymerase. Taq polymerase is the molecule that we're going to be using for PCR reactions. So this is DNA replication in a test tube. And Taq polymerase is special because it was isolated from a bacterium called Thermus aquaticus. Now this is a bacteria that is found in deep sea vents, so it's hot. And that's important because this particular polymerase can withstand really high temperatures. And one of the steps in PCR is denaturing our molecule with really high temperatures. And so normally, if you expose an enzyme or a protein to high heat, you're actually going to ruin it. But Taq polymerase is special and was critically important in the development of PCR because the only way we can reasonably get those molecules apart in a laboratory setting is to heat them up. So just as a quick review, polymerase chain reaction, because I think it's a good sort of introduction to how general replication works. We are first going to denature or pull apart our strands using heat. And then we are going to anneal short little segments of DNA that we have already planned out that's going to help us to isolate the sequence that we want. And we'll actually see an analog to that when we talk about replication in cells in just a minute. Then we are going to actually perform the replication. So Taq polymerase is going to bind and move along, adding nucleotides here to our strand, and that's how we're going to extend our strand. And then we're going to repeat the cycle. We're going to pull them apart, allow our primers to anneal, allow Taq polymerase to do its job replicating the strands, and eventually what happens is we get isolated fragments of just the DNA that we want, and we get many copies. Now this is a little bit different from replication in a cell, because replication in a cell is going to copy all of the DNA and we're only going to get one copy each round. But this polymerase chain reaction is really useful because it can amplify or copy very, very small amounts of DNA. So we don't need a lot of DNA if we're going to do PCR on it, because in theory, you only actually need one strand of DNA. Usually that you need more of it for PCR to actually work, but this is important if you're looking at something like ancient DNA or viral DNA where you don't have a lot of DNA and you're trying to find the DNA. And then it's also important for isolating specific sequences. So if we want to look at specific sequences within a genome, we can just isolate those. And that's, we talked about that a little bit when we were talking about sequencing. All right, so replication is going to begin at what we call an origin of replication. In bacteria, remember, we have a circular chromosome, and bacteria are going to have one origin of replication, so that helicase molecule is going to bind here while it is still closed and pull it apart, essentially making this little bubble where the replication machinery can get in there. And then replication is going to occur in both directions around this bubble, and it's going to extend all the way around until eventually you end up with two identical molecules. So the site that all of this replication begins at is called the ORI-C, or the origin C site in the bacterial DNA. Now, I have this little note here. Do you recall which regions open up easily? Think back to our nitrogenous base pairs. Remember, we have T that makes a double bond with an A and C that makes a triple bond with a G. It's easier to pull apart these double bond TA regions because it requires less energy because we're only breaking 2 hydrogen bonds instead of 3. So these origin sites tend to be AT rich, okay, because it's easy to get in there and pull them apart. In eukaryotes, we have multiple origins. Remember that eukaryotic chromosomes are linear. And so we actually have multiple origin sites all along, and these origin bubbles will eventually sort of merge together. And you end up with, it's not going to be the correct size, but once you have completed all of your bubbles, and all of your bubbles have merged, you end up with your 2 daughter strands. So we're going to focus on bacteria because it is less complicated, but the overall principle is similar in eukaryotes. And so we're not going to worry too much about the differences here. All right, before we can get started on the actual mechanism, I want to give you the functions of some of our enzymes that are involved in this process. So we already talked about DNA helicase. DNA helicase is going to pull apart our strands. Okay, farther down the line, as we pull apart our strands, we are actually going to create tension on our molecule. Now, hopefully I remember to do it. You can demonstrate this, and I like to demonstrate this in lab with a rubber band. So if you happen to have a rubber band, grab a rubber band, and I want you to hold one end, and I want you to pinch the other end, and I want you to twist one end, and you'll see that you make a double helix. You may have already sort of done this. You can see the 2 backbones in there, and you can see the helix. Now, it's hard to do, but if you can hold it between 4 fingers in one hand, hold each end, if you stick your other fingers in between and attempt to open up that rubber band, you're going to get a really, really tightly coiled end. And the tighter the farther you open up this replication bubble, the tighter you're going to make this end of your molecule, because you're putting tension, because if you're holding either end, there's nowhere for that tension to be relieved. And so we have an enzyme called topoisomerase that is going to relieve supercoiling tension. Now this does this by making a single strand break and sort of relieving the stress by allowing the other strand to spin around and sort of unravel some of that tension. So that's really important. If you didn't have that, you would end up with broken DNA molecules. Then we have multiple single-stranded binding proteins. These guys are going to hold the backbones apart so that they cannot re-anneal. We have origin binding proteins. These are the ones that are going to bind at the origin site and help helicase begin to denature. So these are kind of like guiding proteins. We have a clamp and sliding ladder. Now, our book does not give us a picture of this. Other books I've used in the past have given pictures. I'm not super worried about you guys understanding what this looks like, but the clamp loader and the sliding clamp are components that keep polymerase on the strand. Okay, so it helps to actually hold polymerase in place. And then ligase is an enzyme that is actually going to link together our Okazaki fragments, and we'll get to those in just a minute. So you might need to come back and review these once we're all finished. Ligate means to link together or to bind together. So those are the big enzymes, the accessory proteins that are needed for this process. All right, so this is a cool picture here. This is an electron micrograph or an electron microscope image of a replication fork. So here on this end, we have double-stranded DNA. And then here we have single-stranded DNA and the replication fork. So all of those molecules, those helicases and topoisomerases, DNA polymerases, they're all going to be focused around this region. That topoisomerase is going to be ahead a little bit, relieving stress. You'll have your single-stranded binding proteins in that fork, sort of keeping things open. And then, of course, you'll have your big helicase molecule in there actually doing the replication. So in our cartoon picture here, we have the helicase. Helicase is going to move along the strand, and as it does, it's going to open that replication fork wider and wider. So it's going to continue to pull apart these hydrogen bonds. So it would have started back here somewhere and moved along from the origin to the location that it's at now. And then, of course, we've got these single-stranding binding proteins. So first step, helicase comes in, then the binding proteins keep it apart. Then we are going to add in some of these additional molecules. The first one we're going to add in is that polymerase. And the polymerase is going to start adding, it's going to move along this strand and start adding base pairs here, complementary base pairs. So we're going to move sort of at that 3' end toward the 5' end. Now on the other strand, we're going to start at the location closest to the fork, as close as the polymerase can get in there, which is essentially like a 3' end. And then it's going to move this way towards the 5' end, adding the nucleotides. So let's look at some of this in more detail.